of mass education around HIV cure. In fact, if you think about it, there's a lot of people who don't know a lot of stuff about it. And it's our job to really sort of help the people know what's going on. And so that's what you're here for if you didn't know what you're here for. And we're going to do that by showing you 3 different cure modules that community and scientists together have made. There's going to be one that's going to give you a basic overview, and then one that's going to go to children cures, and then one that's going to be close to home and adults, where we're working on a gene therapy approach to curing HIV. So, I want to first go through some things. This is brought to you by the HIV Community Advisory Board, and I am their coordinator, and that is my email. If you want to ever ask me a question, please feel free to do so. I also want to thank all of our collaborators. My drone teacher always said that we should be thankful at the start, so I wanted to say thank you to all of you, especially the AVAC and CARE, who've done an awful lot to bring this here for you. And I also want to thank our sweet library. Thank you so much. Actually, Anne and Jared and Misha, all, I love you. I love that you've allowed us here. So I wanted to say thank you in a very special way, almost like a big to you. There's some other things. CLD News gave us free ad space page in their newspaper and online. I want to thank them. Fred Hutch gave us some sanitizers, and I hope you all got that hand sanitizer. We can benefit for it. And I also want to thank my original place of employment, the AIDS Clinical Trials Unit, for just sort of bearing with me as I sort of grow strange new images and learn how to fly on them. I also wanted to just give you this sort of little heads up. All of these modules that we're going through today are available to you. There's a website at AVAC's, on the AVAC's website. You can see it there, avac.org/core curriculum. You'll be able to use these for your own purposes in terms of teaching other people or reminding yourself, or if you are a teacher, you can use it for your students, or if you are someone who works out in the community in some way, these are for you to reference. This is something that I think is the most exciting part of what we're doing. But I want to ask you first, what have you heard about the HIV cure? I'm sure you all, since you're here, heard something, so find someone next to you right now and tell them one thing that you've heard about HIV cures. Either to your right or to your left. Find it or back and forth. Find someone. I want to see you tell each other something. Introduce yourselves if you need to. Wow. So look at this. You already all know something, or at least you think you know something. So, how many people here heard something from another person that they've not heard before? Who heard anything new, or have you shared information that was already known? Did anyone hear anything new? You never heard? No. Just like back in high school days. Don't raise the hand. Well, I'm going to try to go through some things, and I steal from the best. I learned from Banksy, we learned from Picasso, that's why we need to invent the wheel when you can steal it. So this is actually from a guy named John Mellors, and I thought this image spoke volumes more than any of the science I've ever had to learn about cure. And that's a high jump, and I'm particularly fond of this because when I was a high school English teacher, track season, I ran the high jump. So look at that. That's awfully high. I think that's a perfect metaphor for what curing HIV is going to be. And when Dr. Mellors last year at CROI used this image, it just spoke volumes to me, and it's almost an image that's burned in my head. So the bar is going to be pretty high to get a cure for HIV. Keep that in mind, because it's easy to get discouraged. Also, look at that 4-letter word. And I'm sure your mother told you about 4-letter words, right? That word, you should really be sensitive about using the word cure. And I want you all to just at least realize that we use it often as a sort of catch-all phrase, because it's the easiest way to describe a vast array of things that you're going to be learning about today. But you're going to be learning definitions of the different types of cure we're working on. So I want to give you some definitions of my own, or at least some thoughts about the word cure. First of all, compassion, the C there. We're going to need to cure more than a virus. in a body. We have to cure an entire epidemic that was sort of brought on by the way we live our lives in all sorts of ways. The poverty, the injustice, and the disparities that exist in our lives just prove that. And it's going to take a lot of compassion to heal people fully. And it's also going to take a lot of understanding. That's where I got to you. You really need to understand what's going on. So if you don't know things about bone marrow transplants, or if you don't understand the concept around like monoclonal antibodies, then you're going to have to maybe deepen your understanding a little bit. This is one I always love. There's only one way we're going to get a cure, and it's through research. And I love that research actually has some science now that they can start scientific investigation. But the key is the one that I think is the most important. It's going to take everybody to find a cure for HIV. And I mean that. Whether you are a scientist or a lab rat, whether you are a community person, whether you're a person living with HIV, or you just love somebody who lives with HIV, or love someone who runs around with HIV. It's going to take all of us. It's going to take all of us focusing on a cure and asking people what they need to do to make sure that it comes about in at least my lifetime, because I'm kind of selfish that way. And I would like to leave this earth, see at least the glimmer of the tail of the cure that's going to be coming, if it comes at all. So it's going to take us there. But please keep in mind, there still is no cure to HIV. But here's a little headline for you. How many of you saw this? Raise your hands if you saw this headline. The one where it was saying, Danish scientists saw a cure within lungs. Did you see it? Keep your hands up. Let me see. I want to see. Did you see that headline? Do you remember that? How about this headline? Oh, medical first, a baby with HIV. Did you hear about that one? Yes, the Mississippi baby. How about this headline? 14 HIV patients have functional cure. Did you hear that one? Do you know what country that was in? France. Yes, France. Thank you, Timothy. How about this? Patients, HIV-free. HIV-free after transplant. Did you hear that? Do you know what that one's about? Yeah, no. That's awesome. Good. Bone marrow transplants. Two men in Boston were transplanted, got cured. We'll get to that in a second. How about this one? This one was popular north of Seattle when I went up to Everett for a friendship dinner. Everyone was talking about this one. Bee venom. Did you hear that? This one is just from February 17th. The NIH released some press release that got translated as this. Soon, pill to prevent and cure HIV. What's wrong with this stuff? What's wrong with headlines in general? They turn out not to be true? That could be. Is it that it's not true, or is it something else that's going on? Overly sensationalized? I heard someone. And they're trying to sell you. They're trying to get your attention so they can have commercial space that people are going to pay exorbitant prices for. So they have to sort of get you to buy their paper, go to their website, turn on their TV channel. How about this stuff, though? Mississippi baby now has detectable HIV. Did you hear that? How about this one? The hopes of an HIV cure in the Boston patients are dashed because they both had viral rebound. Or how about this? Your setbacks force HIV researchers to reset. The news is confusing. And if you don't know the science of what we're trying to do, these headlines are going to be sort of like a ship without a rudder. You're just going to sort of be buffeted by all of this news and not understand how things fit in. But wait, it's not even just headlines. Who's that? Magic. Dude. Magic Johnson. Look at that. Cure for AIDS 2013. I figured that leaked out in the 90s. What's that referencing? Have you heard that Magic Johnson was cured? Well, he wasn't, but have you heard that? I just heard it on our Facebook page 2 days ago when I was talking about Timothy's cure, and some young woman said, I thought Magic Johnson was the first person cured. And we have that going on. Has anyone ever heard of Dr. know when I work with a lot of people who are new to HIV, they go from roller coasters of high elation to deep despair. I go to despair sometimes. I hear things. So what do you do to protect yourself? What do you do to protect each other and protect the people who will be harmed by headlines and all of this sort of stuff? Well, I'm going to give you something to think about. First of all, whenever you have any sort of despair, remember the best part of this whole thing is that we now have science going on. Real science. Dealing with the world the way it is that has to follow a scientific method, verify and be true as the rest of human beings and think about that. Science is now going into that pure arena that only those charlatans once ruled. And they're making money still at it, as you can see from my example. But science will take care of this if we can cure HIV. The other thing is, if you ever lose faith, remember that you have one person to cure it already. I mean, I really hold on to that sometimes. If there is one, there must be a chance that there is another. And if you didn't know, Timothy is joining us today. We're sitting over here in Paris, and so feel free to say hi to him. But the one person who has been close to the heart is here in this room with you. And that should mean something. So remember also that cure is in its infancy. It's just a baby right now. What does that mean? Most of those news things, they're still in the lab. They're still in the test tube, or they're in animals. They're not in humans half the time. So when you hear it short within months, but you're still in a test tube, there's a long process that needs to go to get you to where you can say that you have a cure of any sort. And if it is in humans, it's probably a very small study, because we're still worried about safety issues and things like that. So that's something else to keep in mind when you have those headlines bombarding you. This is the last thing that I also saw from John and others, because it's really something that keeps me going. If you ever doubt that we can do this curing HIV, just remember, once in our history, it was probably a fantasy to think that you could take one pill a day to treat HIV. Think of it, back in 1996, if you were telling people that you could take a pill, one pill, and those handfuls of pills that people have to take back then, and sometimes still today. But we have treatment now, one pill a day. And that's a fantasy, that's a reality. So, who knows when the cure is going to come, if it's going to come, but don't lose hope on that. So we have three presenters today. Jeff Taylor, Ann Collier, my boss, and Keon Saucier. And they are going to be going through the basics of HIV cure with you. And I'm going to sort of be moderating this with them. So I think first up we have, I want to give you a special note there. Andrew Johnson is not going to be presenting again. Sorry, Ann, So that's just a little caveat. He's not going to be coming out because they have three of those drawing here. You didn't know. I just wanted to make sure that it's like magic was coming out. So we have this, our basics of HIV cure research. And I think Jeff is the first one who's going to be up. So this is what I'm going to go through. This is what you should get from this lesson. You're going to get some definitions of cure. You're going to get the elements of the reservoir and what a reservoir is. You're going to get the strategies that we are following to cure HIV, and there's four of them, and you should be able to leave this group know that we have four different strategies we're pursuing, and then you should have an understanding of the overall challenges. So, sort of starting with the objective so you know what you should be learning from us today. So, Jeff, you are first.
Thank you, Michael. My name is Jeff Taylor. I've been living 37 years with HIV, and since the beginning to access treatment, I started participating in research trials, including the first ACT trial. So I've been a big proponent of research because it's kept me alive, as we know, it's kept many of us alive over the years, and currently serve on the community advisory board of the CARE Laboratory, which is a sister laboratory for the PHP laboratory based here in Seattle. It's based at the University of North Carolina and focuses on the latency that we'll be talking about later on. So it's a pleasure to be here today and take a look at it. So you've probably seen versions of this picture before. It's a pretty familiar picture showing how the virus gets into a target cell, which is a human T cell, or CD4 cell, and the various steps it takes to make copies of itself and various places along the way where the HIV medicine intervened, be it fusion, integrated into the genome, reverse transcriptase, which of course is a lot of the familiar HIV drugs like ACT, Luvada, and then translation and making cells. So why isn't this enough? We know that if we take our meds, we can stop that reproductive process, and the viral load that we can measure in our bloodstream goes down to 0. But what happens if we stop taking these drugs? We all know the virus is growing back as if we never known before. So the problem here is what we call latency, and that's when the immune system cells that the HIV targets become latent or go to sleep. Obviously, we have all these immune cells in our bodies that are designed to fight infections, but they're not doing it all the time. Otherwise, they feel like crap. Like when you have the flu, your immune system's revved up. You've got these immune system cells activated and actively fighting disease. And one of the side effects of that is, of course, feeling quickly sick. So you don't want that all the time because that can be unhealthy or miserable. So what happens is when the virus is active, it makes copies of itself, and the cell itself dies, and the virus goes on to infect a new cell. However, as I said, sometimes the virus, if it's not exposed to something that it recognizes as something it needs to attack, will go into a rest mode. And this is what we call latency. And when the virus is latent or resting, those cells can't be recognized by the immune system. They're not actively going through that process that we went over the slide, so that the HIV meds can't do anything to it either. So it's basically hiding out. We call these reservoirs, which are hiding places for these latent or sleeping cells. So Michael was talking about cure, and that's, as he said, it's a 4-letter word. It means a lot of things to a lot of different people, and meant different things at different times. Certainly early on in the epidemic, I think many of us remember before we had effective drugs, people were thinking of kind of alternative ways to keep themselves healthy and alive until something better came along. And these were things like herbs, dietary supplements, diets themselves, visualization. A lot of people may remember Louise Hay and Hay Rice. There's lots of positive thoughts. You could keep the disease at bay, and you can ask me later what my opinion of that is. But it obviously was not enough, and people still died. And also, in different cultures, people have different rituals and belief systems that they use to deal with the thought of incurable disease. So certain rituals, prayer obviously is a very powerful therapy for many. And alternative for traditional healers, I think in many cultures, is very important. You see this especially in Asia and Africa, where people turn to their traditional healers to help them with something that they don't have answer to, or that they're afraid that their therapies don't have access to. So it's not a new idea, it's something we've been talking about for a long time. So actually, as a kind of aside to that, I don't know if people remember back in the 80s when AIDS first came out, they said, well, it's a virus. We've encountered viruses before, we have vaccines for virus, and very famously, Jonas Salk, who was the man who developed the polio vaccine back in the 50s, said, I'm going to tackle this, I'm going to take this on. He was at the Salk Institute in La Jolla, next to UC San Diego. And obviously he did it, he died before he was able to do it. We're still looking for a vaccine today. And much of that research is centered right here in Seattle. So it's not an easy thing. They thought that, within a few years they had a cure, we wouldn't be talking about it for years later. So Michael was talking about cures in the media. This is a really famous example. In 1996, when the cocktail first came out, when people first stopped dying, Dr. Hill had this, it was a very rational idea. I mean, now it's kind of silly because it didn't work. But that if people stay on the meds long enough, that the virus would eventually go away. And the problem with that was that they didn't really understand the latency or the sleeping state of the virus that we now described. And obviously that turned out not to be true. The virus was talking about it today. So what does the cure mean? So for those of us living with HIV, obviously what we would like to do another issue, of course, is not being infectious, not being typhoid and giving the virus to somebody else. Being sexually transmitted disease, ironically, the people we love can sometimes be the ones most at risk. So that's something we don't want to have to live with. And then complete viral eradication, getting rid of the virus entirely. Obviously, people don't like to have something you do that's infectious, particularly you or others. People like it gone. There's several ways to look at that. Obviously, there are people who live with the virus who don't get sick, what we call disease controllers, or long-term non-progressors. And then the model for that, we need to remember that HIV didn't just come from nowhere. It came from monkeys and chimpanzees, and they have a form of this virus called SID, or simian immunodeficiency virus. And the natural hosts of these viruses don't get sick. It's only when it crosses species into humans, and we haven't evolved enough yet to live with the virus that they have. So there's a real biological model for that, and one we can perhaps learn from. And scientists, too, don't know what it means or what it should mean. And we've learned this over the years. We don't know how to determine from looking at the lab work what cure might be. And so often we're faced with having to take people off and see about that. Which can be a scary prospect, and it has its own dangers. And that's one of the things that we progress into research that we're going to think about. And really educate people who are making potential volunteers for studies that there are real risks to this, and people have to be really committed to understand what they're getting into. And then, of course, as I mentioned earlier, do we need to get rid of the virus altogether? Maybe we just learn to coexist with it. So that's another approach that we need to be looking at. So there's a lot of different aspects of the research that we need to think about. Pardon us when we figure out stuff.
All right. Thank you. So, is a cure for cancer? Biologically, HIV and cancer can both enter into remission and return from the undetectable level years after treatment ends. And anybody who has ever had anyone die of cancer, unfortunately, this is a reality. You guys know this. I know this. So, HIV and cancer are rapidly mutating and have several different types which can react differently depending on treatment. So, different treatments can produce different mutations. And when those mutations take hold, basically, the disease has changed to something different. And the way that medicines, which we largely use most of the time, I would say maybe 70% of the time to cure things, they're targeted, and they're targeted genetically. So when a genetic mutation happens in a pathogen, that makes it hard to fight. Now the cancer industry uses the word cure in print and online media when they mean remission, okay? And cancer protocols rarely use terminology like eradicate or cure for the simple fact of the matter that remission takes place, that mutations take place. So we're talking about this to prepare people for the fact that if a cure comes, it may be either what's called a sterilizing cure, which is complete eradication of the virus in the body, or, and this is probably more realistic and more likely, it would be a functional cure or remission. So very small numbers of HIV infected cells would remain within the person, and they would stay at undetectable levels or on treatment.
So I wanted to interject here, just because when we had Francoise here over the summer, I think she really boiled that down so simply, where she talked about sterilizing cures just being total elimination of the virus from the body, and she described a functional cure as when the virus is present in the body still, but it's controlled. And so just the way she's kind of made it so simple, it really helped, I think, a lot of people get their mind around it. So I had to throw her up there. Huh? Oh, does everyone know who she is? I just kind of always assumed. She's the woman who discovered the HIV virus in 1983, along with her mentor, Montagnier, and yeah, she got the Nobel Prize. And so she won the Nobel Prize in 2008 for that discovery. Without that discovery, we wouldn't know the virus, we wouldn't have testing, we wouldn't have as quick a grip on the epidemic, which, given the history, might not feel that way to you, but it is remarkable within 30 years how much we've done against one really violent virus.
The other thing about her is she was the head of the International AIDS Society,
Yes.
and she has really done a lot for trying to make sure that HIV research and awareness is worldwide, not just focused in northern countries. And same thing for cure. She's really helped make it a serious scientific objective to have cure.
So, you guys keep on hearing reservoir.
Well, that's me.
That's her.
Okay, so the term reservoir from the medical point of view is really a lot like a water reservoir that we see in Seattle. It's a place where something is hanging out. And so you heard from Jeff about the fact that HIV can get inside of the cell and then just sort of sit there. And what the definition of a reservoir with respect to HIV is, it's where the cell that the virus is hanging out with, presumably doing nothing, are located. And this diagram shows some of the places that it is currently believed that the cells that HIV live in hang out. So the brain is one of them, the intestine, and in particular there are what are called lymphoid pockets. There's sort of little immune cells, the body's defenses, that hang out in the lining of the Gowdy intestine, actually. There is something called the spleen, which is an organ in the belly that acts like basically like a sponge for blood. And there are a lot of HIV cells that can be there. And there are probably places in the body where HIV is hidden inside itself that we don't even know. The studies have done most work with blood because it's the easiest tissue to get without doing invasive procedures. So we know that there are latent cells in the blood, although they're very, very, very rare. And there's a lot of research being done to try and sort out where the other cells that the virus can help in are. And you heard about T cells. There's another kind of white blood cell called the macrophage, which can carry HIV and actually is thought to be like a Trojan horse, carrying HIV early on into the brain. So there is an avenue of research to really kind of understand these reservoirs, where they are and the significance, and what we can do to get rid of them. So I said we're not even sure where they are, so how is the reservoir maintained? Well, that's really not known either. It shows just how many questions need to be addressed to try and cure HIV. So you know that some cells have this property called latency, that they're sort of think doing nothing, perhaps not even having something on their outside that shows they have HIV. And so while there are theories about what keeps those cells hidden, it's not completely clear yet what those factors are. There are ideas about them, but at the moment there are more theories than there are facts with respect to how the reservoir actually maintains itself. The term active reservoir refers to cells that are capable of producing the virus even in the presence of effective antiretroviral therapy. So current antiretroviral therapy works by shutting off the cell's ability to help HIV make more HIV. But there are some systems in the body that have been shown where even in the presence of effective HIV, a cell that has HIV in it can make another one of itself. And so And at the moment, it seems that most of the tests that are used that either measure teeny amounts of viral RNA or the DNA that HIV has inside the cell, while they're associated with each other. So if somebody appears to have more RNA in the cells or more DNA, they tend to have more on both tests. But none of them seem to measure the same thing, and none of them are very easy or cheap. So there's a lot of interest right now to try and figure out what a better test might be than the ones that are currently available. What we do know is, using the tests that are available, the amount of this latent virus that people have, it can vary greatly. When people have been diagnosed late in their infection, so they've had HIV for a long time, not on treatment, we know they tend to have larger reservoirs than individuals whose HIV was diagnosed very early after they got infected. The other thing, and I did say something, I did mention that the cells are hanging out, supposedly doing nothing, but they're really not doing nothing. Because what we know is that when HIV is infected cells in the body, they have the ability to make the immune system more active than it is normally. And that, when you may say that's really weird, I heard that HIV makes the immune system suppressed, which is true. It makes it very suppressed. That's the active replicating cells that do that. However, the cells that are hanging out, hiding with HIV in them, also have an effect on the immune system, but it's activating parts of the immune system. And what we know from research, from a lot of research done, not just for HIV, but for other things, is that when certain parts of the immune system are activated, that can cause damage. And especially to certain kinds of the body organs, like the heart, and the brain. And so the notion is that, unfortunately, even in people who take HIV medicines and have their suppressed virus, some of them have more evidence of this inflammation or immune activation than if they didn't, if they were compared to people who don't have HIV. So unfortunately, probably reservoir size does matter for overall health. And certainly with respect to the cure part, you think it would be harder to cure someone who has a big reservoir.
Let me interject here. How many of you have heard of reservoirs before? Well, that's pretty good. I have a friend of mine who never heard of them until I showed him this slideshow. And he's been infected for 20 years, and it just makes me realize how much that I take for granted. And I just wanted to see if this was a common bit of knowledge for our audience here or not. I just was very sort of puzzled and trying to explain it all to him yesterday. It's just really amazing for me to experience. Sorry for that interjection.
I did mention that if people are recognized to have HIV right after they're infected and treated, they may have a smaller reservoir. And there are some data to suggest that. There is a group in Thailand who have studied a very large number, actually almost 80,000 people who got HIV tested, who didn't have HIV, who got tested every 3 days. And some of whom acquired HIV during that period, and then were obviously only had it for a few days. And by studying these volunteers, it's been shown that early treatment can help markedly decrease the size of the reservoir. And it's possible that if you decrease the size of the reservoir, there may be places in the body that don't actually get HIV in certain kinds of cells. But that's still something that's being studied, and how early, obviously, most people in the rest of there is another such study that is being looked at in South Africa, but they have mostly been looking at trying to educate folks rather than doing early treatment. Now, there are some very rare individuals who actually do have natural immunity to HIV. And although most of them are of European origin, if I make a slide, I would not say that they were all, but they're often, very often, but who knows, there may actually be some individuals who aren't from North America who have this genetic change in their cells. And the change that I'm referring to is a protein on the outside of a certain kind of white blood cell. Those kind of white blood cells are called CD4 cells, and those are the major target of HIV. But for HIV to get into a cell, it has to not only be attached to this CD4, it has to also attach to something called a co-receptor. And there's another protein also on the outside of those cells. And we do know from other studies, even before HIV was recognized, that there are a small number of people who have a mutation in this protein called the CCR5 receptor, where they have just a little bit of a change in it. And if you happen to have CD4 cells that don't have a regular CCR5 receptor on it, that cell, most HIV can't get into that cell. So there are some people who have two copies, one from each parent of this change, and those are individuals for whom it is thought to be almost impossible to have HIV get into their cells and therefore can't get infected.
So, I think that it's so good in the background. I think it's more complicated.
And the question is, have they been able to sequence the amino acids that are changed? And the answer is yes. So it is known exactly what change in the protein has to happen in order to render resistant to HIV. So that is known, and we're going to be talking a little bit later about stem cells and Timothy's stem cell, which actually took advantage of this very piece of knowledge. What we do know is that the people who have this deletion in CROI generally are very healthy. They have a little bit of trouble against certain pretty rare infections, but otherwise they seem to live a normal lifespan, and it doesn't seem to impact daily. back up. A long-term non-progressive. Maybe you just kind of give me a speed up. I can. And those are people who have HIV but who have normal CD4 counts. And I mentioned that the CD4 cells are the ones that HIV wants to infect. And so these and CD8 cells are some other cells in the system that help kill things. But long-term non-progressors are people who have HIV but whose body defenses have not resulted in or HIV has not resulted in the death or the decrease in the number of those cells, despite not having them. So those people have also been studied for the same reason, trying to figure out what's different about their immune system or their virus. And unfortunately, there doesn't seem to be any one thing that has led to long-term non-progressors. There are a lot of different things. But the idea is if we understand more about those, that may help us still understand how to better keep HIV under control if we're good. I think that's fine. Is that right?
Yeah, so we are going to be pressed for
Here.
time.
I'm
Yes.
sorry.
So, no, you can still go on. I just wanted to let you all three know, so we try to bring this in. So go on.
All right, so what are the scientific challenges to finding a cure? Because we all know that while science says magical things, it's not magic. So, I understand the latency pathway of HIV. Why do the cells remain latent? I think that's going to be one of the biggest challenges, finding that out. How does the reservoir persist over our lifetime? Why does it increase? What, if any, effect does sex have on latency? Factors still known and under debate. Determining the location of latently infected cells, e.g., is it the gut, is it the T cells? Where exactly are these targeted latent cells? And then finally, measuring the size of infected reservoirs. So current pure research strategies are kick and kill, gene therapy / gene alteration, stem cell transplantation, which we have Mr. Brown as an example of, and therapeutic vaccines. So, what is kick and kill? The strategy aims at forcing cells out of the resting state so that the virus can be released and killed. So basically, once the cells start replicating, again, they can be identified and killed. So, giving a massive shock to your body, waking up the virus, getting it active, and then having some agent to come in and eradicate it. Okay? Virus is basically released in the bloodstream and affects cells, however, effective ART, which is antiretroviral therapy, blocks replication. However, a kill component like a therapeutic vaccine or some kind of medicine would be needed to eliminate the virus. Okay?
Well, challenges. What are the challenges that you feel?
Not every infected latent cell will become active with latency reversal agents. So that's one of the challenges. It's been shown that a latent cell may need several rounds of stimulation to start purging the virus. So basically meaning that, if you're revving up that virus and attempting to eradicate it, but nothing's happening when you're revving up the virus, that's going to be damage to the body. And then finding effective latency reversing agents that actually stimulate the cells inside the body. Okay? And this is kick and kill in action, basically. HIV, DNA. Just like that. No, it's not.
Sorry.
So it activates, and it turns on the RNA, the DNA, and the proteins of the HIV. Okay? And the HIV virus and whatever agent is introduced causes cell death. So this is that in action.
Okay.
So gene therapy, I was talking about gene therapy, or altering our genes. And then the aim here is to remove a key element that the virus needs to invade the cell. And as Dr. Collier was talking about, the CCR5 receptor is one of the necessary binding sites for the HIV to get into that cell. So the idea here is that if you knock out the genes that create that CCR5 receptor on the outside of your target cells, those cells become resistant to HIV. And why do we know this? Well, again, they did this through a stem cell or a bone marrow transplant. And you'll hear more about that later and why the average person doesn't want to do this because they wouldn't want to. So scientists are working on ways to modify culture and introduce these revised or altered cells into the body of patients who increase resistance to HIV. So you take a normal person living with HIV and you collect cells from them. Normally this is done by blood collection, and they filter out the target cells usually T cells or CD4 cells, sometimes stem cells. And then they edit them. And what they do, the best way to describe this is they go in with a little pair of what they call molecular scissors, and they very carefully target and snip out the genes that are responsible for creating that CCR5 receptor on the outside of the cell. So you cut that out, and then you create cells that don't have it, and therefore they are resistant to infection. So then they take these modified cells, they put them in a test tube or petri dish, and they grow them to make more. Because you can only harvest so many per person. You don't want to take everything out. So the idea is to create as many as you can, and then put them back in those revised HIV-resistant CD4 cells or stem cells back into the patient, and that should make them resistant to HIV. So that, in a nutshell, is kind of how it works. So what are some of the challenges? Well, that doesn't kill the existing virus in the body. It just alters your cells so those new cells are HIV resistant and are better at fighting, better at using new cells. And I think, we all know about Darwinism and the process of selection. What happens is those new improved HIV resistant cells are the ones who live, and if you go off your HIV therapy, the other ones get killed off. And so eventually you have a population of newly established gene-modified HIV-resistant cells in your body that can resist HIV over time. So another problem is that this is tricky business. When you start going into the human genome and mucking it up, bad things can happen. Of course, cancer is one of those. So you have to be really careful that what you're doing is very precise. You're only targeting the genes that you need to take out, and there's no what they call off-target effects, where you're being imprecise and maybe affecting other genes that could do bad things like cancer.
So basically, we don't want to try and cure HIV and create a zombie virus.
The zombie apocalypse, is that what we're looking for?
So another potential way to cure HIV, which it did, is to do something called stem cell transplantation. So stem cells are the cell in the body that are the very, very early cell that then can become lots of different kinds of cells. So a stem cell transplant is done by taking these cells out generally from babies or from someone else, but in order to give stem cells to another person, you have to get rid of their immune system. So someone who has a stem cell transplant usually ends up with chemotherapy, radiation therapy, and then is like at great risk if they don't have defenses against anything. And that wiping out process is called conditioning. So it often, so it's a very, very vigorous thing to undergo and not pleasant, but it is necessary in order to then be able to give someone stem cells. So what's on the lower right-hand side of this is just illustrates the fact that these stem cells can go on to make lots of different kinds of cells. So what was done in a brilliant way is if the donor for the stem cells has the mutation for this CCR5 receptor, so those cells can't get infected with HIV, like was done for Mr. Brown, then if that transplant takes, then HIV couldn't enter the cells that then populate the body. And so that's why it's a really incredibly smart strategy. Unfortunately, it does have challenges because with the new immune system, there is a possibility that the new immune system, which is called the graft, can attack the old immune system, which was supposed to be gotten rid of. But, so there's this thing called graft versus host disease, which can make people who have transplants be sick. In addition, the conditioning is really dangerous, as I said. And there are very, very few people who have this mutation, so there's not a lot of donors to be able to use this. And it's very, very expensive, so it's not a realistic thing that we could use to help cure lots of people with HIV, even though it was successful for one wonderful person who's helped energize this field.
And we're going to be bringing Timothy himself, as well as the doctor who did that experiment, here to the library next Thursday at 6 p.m. and giving you a chance to ask the man, both of them, whatever you want to ask them. So it's a rare chance, something that I've been looking forward to for a long time, and I'm glad I can bring it here. Neil?
So then finally, there are therapeutic vaccines, and they strengthen the immune response to enable the destruction of newly infected cells in order to achieve a functional cure. All right? So lead controllers are and have been studied for many years to determine if there's a unique biology that might have applications for the general HIV community. Now, the challenges are, of course, HIV mutates very rapidly, as I already earlier indicated. It can be difficult to find a broadly neutralizing antibody that can effectively work long enough to knock out all of HIV. So, if we do come up with a vaccine, it would be, at this stage, for a type of HIV. HIV has different types of licensing mutations. All right, it may not work for all of them. And then overstimulation of the immune system can lead to an increase in cardiac cell survival. So once again, kind of visiting back to the pick and kill issues. You start turning things on in the body, sometimes things may not go the way you think. So the general challenges to finding a cure are animal models. Unfortunately, and sorry if it is part of PETA and stuff, because I love animals, I love chimpanzees and stuff, but before we do anything with people, we gotta do stuff with them. We need better animal models to predict the likelihood of side effects, as well as the effectiveness of new drugs, as awful as that is. Unfortunately, that's unfortunately how the game's being played right now. If they can come up with something else, that'd be great. Participant risk benefit. So unlike treatment, most people living with HIV have effective regimens that work with their daily routines. And not all cure strategies will be worth the risk to city participants. So if you're doing well, you're taking your meds every day, there's nothing wrong with you, you're not checking out, and you're not in danger of checking out. Someone wants to use you as a safer cure, turn things on that may inadvertently get you sick, and you're worse off for the fact. It gives a lot of people pause. Why would I enter this and go through all of that when there's nothing wrong right now? If it's not broke, don't fix it, and a lot of people prescribe to that when it comes to things like HIV. Low capacity. There are very few labs in the world that have the technical equipment and personnel capabilities to conduct cure research. And that's just a fact. And then finally, knowledge. Need for greater understanding of HIV latency in both infants and adult populations. So this whole disease, a lot of it, a lot of our understandings, a lot of our stigmas, and a lot of the way it's been spread is because of misinformation. So once again, it's being on board, making sure we have the right information before we go ahead and do this.
So I just wanted to kind of end it up here, like that high bar that I talked about. You can kind of get a sense that it's really high, but you know that the exciting part is that we can actually start working on this. And there's a whole young squad of these junior investigators that I've met, and there's been these kids that are in undergrad programs that are saying they want to be HIV researchers. I couldn't believe it, but you know, there's hope in that we have science there. We have scientists that are going to work on these challenges. But we need more research in all of those areas. And the good thing is that it's being funded. Your research is being funded by the federal government, and it's also an international effort. So with all of those brilliant minds around this earth working on it, I'd like to dream that impossible dream, if you know Nana Mantra or whatever. It's one of those songs that I sort of feel like right now is there. So I wanted to say thank you for sitting through this first part. It's a little